entirely about growth and repair. It's basically cellular cloning.
Cloning, yeah.
You end up with two genetically identical daughter cells, keeping that full duplicate count, $2. Just goes to $2. It's like a copy machine.
Okay.
Meiosis, though, is completely different. It's an engine of transformation. Its function is gametogenesis making sex cells, and the outcome is 4 genetically unique daughter cells, where the chromosome count has been successfully cut in half. $2 goes to no one.
So reduction and randomization.
Exactly. It's a calculated reduction in randomization.
And these very different outcomes are completely dictated by how the process works, right? Especially how the DNA is handled. We know both start the same way. DNA gets replicated once before any division begins.
That initial replication is common to both.
But that's where they really diverge, isn't it?
Dramatically. Mitosis just proceeds with a single division. That's it. Meiosis, however, undergoes 2 sequential divisions. 2 rounds. But the most crucial difference, the real why behind all that diversity, comes down to 2 things that only happen in meiosis the first. First, you get this tight physical pairing of homologous chromosomes, the ones you got from mom pairing with the ones from dad. That's called synapsis.
Synapsis, okay.
And that forms these structures known as tetrads because there are 4 chromatids involved. Second, meiosis absolutely requires crossing over.
Ah, the shuffling.
The shuffling, the physical exchange of genetic material between those paired chromosomes. Mitosis does neither of these things. It doesn't pair homologs like that, and it definitely doesn't cross over.
So mitosis just lines them up, splits them once. Meiosis lines them up, shuffles them, then splits them twice.
That's a great way to put it, a much more involved process.
So these 2 divisions are needed to get that final reduction. Which division actually does the heavy lifting of cutting the chromosome number? And what's the point of the second one?
Well, the sources define this very clearly.
Yeah.
Meiosis, the first, is the reductional division. That's the key one. This is where the homologous chromosomes separate.
The pairs split up.
Right. Think about it. You have a pair, one from mom, one from dad. In meiosis the first, these 2 homologs physically pull apart. That's the moment the chromosome number is officially halved.
Yeah.
You go from 2 down to another.
Even though the chromosomes themselves are still kind of doubled up.
Exactly. They're still duplicated, made of 2 sister chromatids. But the sets of chromosomes have been separated.
Got it. Reductional.
Then you move into meiosis the second, which is the equational division. This phase is much more like a standard mitotic division. Its only job, really, is to separate the sister chromatids, those 2 identical halves of each duplicated chromosome that are still stuck together.
So it's called equational because the number of chromosomes per cell doesn't change during meiosis the second. It just splits what's already there.
Precisely. You start meiosis the second haploid, and you end haploid. You've just separated the duplicated copies into individual chromosomes.
Okay, let's really focus in on meiosis I because, like you said, this is where all the genetic chaos, well, the productive chaos gets introduced. It all kicks off in prophase, which the sources say is the longest and most complex phase of the entire cell cycle.
It really is incredibly complex. During prophase I, that replicated DNA, which starts as a sort of tangled mess of chromatin, condenses down into distinct, visible chromosome structures.
Yeah.
And then the crucial event happens. Homologous chromosomes find each other.
They pair up.
They locate each other and pair up perfectly, side by side, gene for gene. This tight pairing, synapsis, forms those bivalents, or, as we mentioned, tetrads, because you've got the 4 chromatids all lined up.
That sounds incredibly precise. How does the cell make sure they're held together tightly enough to actually swap bits? Does it need some kind of scaffolding?
It absolutely does. That's where the molecular architecture comes in.
Yeah.
The sources really highlight the synatomal complex, or SC. You can think of the SC as this highly specialized, protein-based zipper or scaffold. It forms right between the 2 homologous chromosomes and locks them together in this incredibly stable, tight configuration.
Like zipping them up side by side.
Exactly. And this precise alignment isn't just structural, it's the absolute requirement for the critical exchange that happens next. Crossing over.
Ah, crossing over. That's the key to recombination, right? Where the actual shuffling happens.
That's it, precisely.
Yeah.
Crossing over is the physical event where non-sister chromatids, so one chromatid from the paternal chromosome and one from the maternal chromosome, literally exchange corresponding segments of DNA.
They break and swap.
They break, swap pieces, and then rejoin. This exchange happens at specific spots called chiasmata, which you can sometimes even see under a microscope as these cross-shaped structures.
Wow.
And the resulting chromosomes, they're now recombinant. They carry a novel mix of maternal and paternal DNA that didn't exist before, massively increasing genetic variation.
Okay, so doing that break and repair sounds delicate. You need some pretty specialized tools to make sure it happens correctly, I imagine.
You definitely do. It requires atomic precision. The cell uses a highly specialized team of recombinase enzymes. The sources specifically mention SPO1. Think of SPO1 as the molecular scissors.
Okay.
It initiates the whole thing by making a controlled double-strand break in the DNA. That break is needed to start the exchange. Then other proteins like RAD51 and DMC1 jump in. They're essential for finding the homologous sequence on the other chromatid and ensuring the break is repaired accurately, using the other chromatid's DNA as a template.
So it's a controlled break and a controlled repair using the partner chromosome.
Exactly. And if this team messes up, if the repair isn't clean, that's when you get those structural errors like bits being deleted or stuck in the wrong place, which we'll talk about later when we discuss diseases.
Right, okay. So while all this intense DNA shuffling is going on, the rest of the cell is also getting ready, isn't it? Big organizational changes happening.
Oh yeah, the whole cell is restructuring. The nucleolus, that little dense spot in the nucleus, fades away. The nuclear envelope itself fragments. Our sources say it actually merges briefly with bits of the rough endoplasmic reticulum.
Huh, interesting.
And crucially, the spindle microtubules start to form. These are the protein ropes that will eventually pull the chromosomes apart. They originate from the centrioles, which have that really intricate $9 times 3D3 arrangement of microtubules mentioned in the notes. It's amazing structural complexity just to get ready to pull things apart later.
Okay, so prophase I wraps up with shuffled chromosomes and the cell ready to divide. Then we hit metaphase I.
In metaphase I, those tetrads, the paired homologous chromosomes, now potentially carrying swapped segments, get pulled and pushed until they line up along the metaphase plate right down the cell's equator.
And this is where it gets really interesting, you said earlier. This alignment is where independent assortment comes in.
Yes, independent assortment is. Well, it's a concept that truly showcases the randomness built into meiosis. It simply means that the orientation of one homologous pair, like whether the maternal copy faces north and the paternal copy faces south, or vice versa, is completely random and independent of how any other homologous pair happens to line up.
So each pair lines up randomly, regardless of the others.
Totally random. It's like flipping a coin 23 times, once for each pair of human chromosomes. Heads it faces this way, tails it faces that way.
And the statistical result of that.
It's staggering. This is where we really need to pause and appreciate the scale. The mathematical potential for unique combinations just from this stat is $2 a runner, where n is the number of chromosome pairs.
Okay, so for humans, n is 23.
Right, so $223, that comes out to approximately 8.4 million.
Wow. 8.4 million possible genetic combinations just from how they line up.
8.4 million unique combinations possible in a single sperm or egg cell. And that's before you even factor in the additional variation from all the crossing over events that happen back in prophase I.
That's incredible. So take that 8.4 million possibilities from one parent, combine it with 8.4 million from the other. The chances of two siblings, unless they're identical twins, being genetically identical are basically zero.
Practically zero. It really highlights why siblings, even with the same parents, can look and be so different. They essentially won different genetic lotteries, thanks to independent assortment and crossing over.
Okay, so after they line up randomly, the cell moves into anaphase I. This is the separation part.
This is the decisive step, yes. Anaphase I is where the reduction is actually executed. The homologous chromosomes separate, the pairs break apart.
Not the sister chromatids yet.
No, absolutely not. Just the homologous pairs. They begin moving toward opposite poles of the cell, pulled along those spindle microtubules by sophisticated motor proteins like dynein and kinesin. They literally walk along the microtubule tracks.
We mentioned earlier that this complexity makes meiosis fragile. The source material highlighted a really crucial molecular protection mechanism here, didn't it? Something to prevent it all falling apart during this reduction step, specifically about the sister chromatids.
Yes, this is a really elegant piece of molecular engineering. Remember, sister chromatids, the identical copies made during DNA replication, are held together along their entire length by a protein complex called cohesin. Think of it like molecular glue or handcuffs.
Okay, cohesin holds sisters together.
Right. Now, normally, to separate chromosomes or chromatids, an enzyme called separase gets activated, and its job is to cut or cleave this cohesin.
So separase breaks the handcuffs.
Exactly. In anaphase I, separase is activated and it does cleave the cohesin, but only along the chromosome arms. This allows the homologous pairs, which were held together at chiasmata on the arms, to separate.
But not at the center.
Critically, not at the centromeres. The cohesin located right at the centromere, that central constriction point holding the two sister chromatids tightly together, must be protected during anaphase first.
Why? What would happen if it got cut then?
Catastrophe. If that centromeric cohesin were cleaved in meiosis I, the sister chromatids would separate prematurely. They go to opposite poles along with the homologs. You'd end up with completely messed up chromosome numbers in the very first division. Massive non-disjunction.
So what's the shield? What protects that specific cohesin at the centromere?
That protection comes from a specialized protein called shugoshin. It's a great name. Apparently it translates roughly to guardian spirit or guardian angel in Japanese.
Guardian angel. Okay, that fits.
It really does. Shugoshin specifically hangs out at the centromeres during meiosis I and shields the cohesin there from being cut by separase. This ensures the sister chromatids stay firmly joined together. They have to stay together for meiosis II to work properly.
That's absolutely critical then. Shugoshin is the key to getting through meiosis I correctly.
Non-negotiable for success.
Okay, so homologs have separated. Finally, we reach telophase I and cytokinesis.
Right. The separated homologous chromosomes arrive at the opposite poles of the cell. In some species, the nuclear envelopes actually reform around these chromosome clusters. And crucially, at this stage, the cell is officially haploid, N.
Haploid, even though the chromosomes are still doubled.
Yes, because each new nucleus only contains one set of chromosomes. Example, one copy of chromosome 1, one copy of chromosome 2, etc. Even though each of those chromosomes still consists of two sister chromatids. Then cytokinesis usually follows immediately, dividing the cytoplasm. In animal cells, that happens via an actin-myosin ring pinching the cell into the cleavage furrow. In plants, they build a cell plate down the middle.
And that concludes meiosis I. The reduction division is complete.
Reduction complete. We now have two haploid cells ready for the next step. All right, now we transition into meiosis II. If meiosis I was the really specialized high-risk reduction step with all the pairing and crossing over, meiosis II is often described as more of a cleanup phase.
Cleanup phase, how so?
Well, structurally and mechanically, it's very similar to mitosis. The big difference is we're starting with cells that are already haploid. They only have one set of chromosomes, though remember, those chromosomes are still duplicated.
Okay, so if the reduction already happened, what are the main goals or, I guess, the risks still involved in meiosis II?
The main risk is still non-disjunction, the sister chromatids failing to separate properly this time. But the primary goal is simply to finish the job. Separate those sister chromatids that have been stuck together since DNA replication.
Got it. So it starts with prophase II.
Yes. In prophase II, if the nuclear envelope reformed in telophase U, it fragments again. The spindle microtubules quickly reassemble in each of the two cells coming out of meiosis I, and the chromosomes condense a bit more.
And then metaphase II, how does the alignment compare to metaphase I?
That's the key difference you can see. In metaphase I, we had the homologous pairs, the tetrads, lining up. In metaphase II, the individual duplicated chromosomes line up singly along the equator of each cell.
Okay, so not pairs, just single file lines.
Exactly. This arrangement looks structurally identical to how chromosomes line up in mitosis.
Which then sets the stage for anaphase II, the final separation.
This is it. This is the moment the cell makes the final cut, you could say. That Shugoshin protection, which was so vital back in anaphase I to keep sister chromatids together, it's now removed.
The guardian angel steps aside.
It does. This allows separase to finally cleave the remaining cohesin right at the centromeres. And now the sister chromatids finally separate. They're pulled towards opposite poles of the cell.
And once they separate, they're considered individual chromosomes, right?
That's correct. As soon as they separate, each chromatid is now considered a full-fledged, non-duplicated chromosome.
So what's the final head count after all this?
Telophase II wraps everything up. New nuclei reform around the single set of chromosomes gathered at each of the four poles, 2 poles in each of the 2 dividing cells. The chromosomes start to decondense, relax back into chromatin, and cytokinesis divides the cytoplasm one last time.
And the result?
The definitive non-negotiable result of this entire intricate 2-division process is 4 haploid, genetically unique cells. Each contains a single set of non-duplicated chromosomes. These are the functional gametes, sperm, or eggs ready for whatever comes next in the life cycle, like fertilization.
Okay, let's step back for a second. It's clear this whole system is just a masterpiece of generating controlled variation. Can we synthesize the main ways diversity gets introduced, going beyond just the mechanics we discussed?
Yeah, we can really boil it down to 4 main sources of genetic variation. The first 2 are built right into meiosis itself. Number 1, crossing over back in prophase I.
The physical swapping.
Right, where segments are physically swapped between homologous chromosomes, creating those novel recombinant chromosomes that are a mosaic of maternal and paternal DNA.
And number two?
Number two is independent assortment in metaphase I, that random alignment of homologous pairs. That's what generates those, massive 8.4 million potential combinations just based on which way the pairs face.
Okay, so crossing over and independent assortment are internal to meiosis. What about the other sources?
The other half involves, you could say, external factors or chance. Number three is random fertilization.
Right. Which sperm meets which egg?
Exactly. We already talked about the 8.4 million possibilities per gamete. When you combine the random output of one parent's meiosis with the random output of the other parent's meiosis, the potential diversity in the resulting zygote becomes practically infinite. It guarantees massive genetic shuffling within the population.
Ensures everyone, except identical twins, is unique.
Fundamentally unique. And finally, number four, we have to include mutations.
Ah, the ultimate source of new stuff.
Right. While they're relatively rare and often harmful spontaneous changes in the DNA sequence, whether tiny point mutations or bigger structural changes, are the ultimate raw source of completely new genetic information. If these mutations happen in the germline, the cells undergoing meiosis, they get passed on. They provide the novel variations that evolution can then act upon over time.
Okay, so you have these mechanisms generating variation, but given the sheer complexity, two divisions, precise pairing, swapping bits of DNA, selectively cutting proteins like cohesin, the whole thing must be under incredibly tight molecular control, right? There must be a master clock making sure it all happens in the right order.
Absolutely. It's exquisitely orchestrated.
Yeah.
The primary regulators of the cell cycle and therefore meiotic progression are enzymes called cyclin-dependent kinases, or CDKs.
CDKs, yeah.
But they're inactive on their own. They only get switched on when they bind to their partner proteins, the cyclins. For meiosis, the key players are primarily cyclins A and B. These CDK cyclin complexes act like molecular switches, pushing the cell past critical checkpoints and into the next phase.
So specific complexes trigger specific steps?
Exactly. For instance, the actual decision to, say, leave an arrested state and enter meiosis is specifically triggered by a complex known as MPF, maturation promoting factor. That's basically cyclin B bound to CDK1. MPF gives the go signal for the cell to commit to meiotic division.
Okay, so that handles the timing, the progression. But what about quality control? Meiosis is so high stakes. If a mistake slips through, it can be permanent for the offspring. Does the cell have a way to, check its work?
It absolutely does. It has to. This is handled by what we call the integrity checkpoint system. It's an elaborate molecular surveillance network that's constantly monitoring the success of key steps, especially that complex pairing, synapsis, and recombination during prophase I.
So it's checking if the chromosomes paired up right and if the crossing over happened cleanly?
Precisely. The cell is highly sensitive to DNA damage, especially breaks that aren't repaired properly. The sources really emphasize the role of the p53 family of proteins here. If severe irreparable damage is detected, particularly maybe during the G1 interphase before meiosis even starts, the p53 pathway acts as a critical decision point.
What does it decide?
It can decide to trigger apoptosis-programmed cell death, basically a cellular self-destruct mechanism.
Ah, so it sacrifices the potentially faulty cell.
Yes. It ensures that severely damaged germ cells are removed from the pool, preventing potentially harmful mutations or chromosome errors from being passed on to the next generation. It's a crucial protective mechanism for the species.
Okay, this brings us directly to the connection between this beautiful, complex molecular process and its real-world consequences. Our sources are incredibly clear on this. Errors in meiosis, despite all those checkpoints, are the single largest known cause of congenital and chromosomal disorders.
That's right. The very complexity that generates diversity is also what makes it inherently fragile and prone to error.
And what's the main type of error?
The primary mechanism of failure, the one that accounts for the vast majority of chromosome abnormalities, is non-disjunction.
Non-disjunction, what does that mean exactly?
It's simply a failure of separation. It means either the homologous chromosomes fail to separate properly in meiosis the first, or the sister chromatids fail to separate properly in meiosis the second. They don't disjoin, they don't move to opposite poles like they're supposed to.
And does it matter when that failure happens, meiosis extensa versus meiosis the second?
Oh, it makes a huge difference in the outcome. This is a critical distinction highlighted in the source material. If non-disjunction happens in meiosis, meaning the homologous chromosomes stick together, then both cells entering meiosis the second will have an abnormal chromosome count, one with an extra chromosome, one missing one.
Ah, so the error propagates.
Exactly. And after meiosis the second finishes, all 4 resulting gametes will be aneuploid. They'll have an abnormal number of chromosomes. Usually 2 will have an extra chromosome and 2 will be missing one. This tends to be the more severe scenario in terms of outcome.
Okay, what if the error happens later in meiosis the second?
If non-disjunction occurs in meiosis the second, so meiosis I went fine, but then in one of the cells, the sister chromatids fail to separate, the error is less pervasive. In this case, the other cell from meiosis, the thacket, divides normally, so you end up with 2 normal euploid gametes and only 2 aneuploid gametes, one with an extra chromatican chromosome, one missing one.
I see. So half the gametes are still normal if the error is in meiosis the second.
Correct. Knowing whether the error likely occurred in meiosis the first or meiosis the second can actually be important for genetic counseling and understanding recurrence risks.
And these failures, these non-disjunction events, directly lead to what are called aneuploidy syndromes, right? Where the fertilized egg ends up with the wrong number of chromosomes.
Exactly. The most common result is a trisomy, where there's an entire extra chromosome or sometimes a monosomy, where one is missing.
Trisomies are probably the most well-known.
They are. And the classic example is Down syndrome, which is trisomy 21, caused by having 3 copies of chromosome 21 instead of the usual 2.
And that seemingly small extra bit of genetic material has huge effects.
It does. It creates a gene dosage imbalance, 3 copies worth of protein products instead of 2 for all the genes on chromosome 21. This disrupts the complex, coordinated ballet of development, particularly affecting the brain and heart. This leads to the characteristic features, intellectual disability, congenital heart defects, and interestingly, a significantly increased risk for things like early-onset Alzheimer's disease later in life.
And trisomies involving the larger chromosomes, they tend to be much more severe, often lethal.
That's generally true, yes. Larger chromosomes carry more genes, so the dosage imbalance is much greater, causing more profound disruption. Edwards syndrome, which is trisomy 18, involves 3 copies of chromosome 18.
Okay.
This leads to severe developmental problems like micrognathia, a very small jaw, serious heart defects, clenched fists, and sadly most infants don't survive past the first year. Similarly, Patau syndrome, trisomy 13, results in major midline defects, things like cleft palate, problems with brain formation, holoprosencephaly, extra fingers or toes, and microcephaly. It's also typically incompatible with long-term survival.
It really underscores how incredibly precise the dosage of genes needs to be.
Absolutely. Just 1 extra copy of even a medium-sized chromosome can be devastating.
It's interesting, though, that errors involving the sex chromosomes X and Y often seem to be less severe than errors involving the autosomes, chromosomes 1 through 22. Why is that?
That difference largely comes down to a process called X inactivation. The body has a natural mechanism to silence most of the genes on one of the X chromosomes in females, XX.
Ah, the dosage compensation mechanism.
Exactly. So take Turner syndrome, XO. This is actually a monosomy X. The individual's missing a second sex chromosome, often lost due to non-disjunction during meiosis in either parent.
So just one X.
Just one X. While individuals with Turner syndrome do face challenges, short stature, a webbed neck, potential heart issues like aortic coarctation, and infertility, it's generally survivable, likely because the body's already geared to function largely with 1 active X chromosome.
Okay. And what about having an extra sex chromosome like in Klinefelter?
Right. Klinefelter syndrome, XXY, involves an extra X chromosome in males. Again, because of X inactivation, the body can often silence the extra X.
But maybe not completely.
Not always completely or uniformly, which is why there are still effects. Individuals with Klinefelter are typically tall, often infertile, may have some breast development, gynecomastia, and might experience mild learning difficulties. But compared to autosomal trisomies, the effects are generally less severe because the system has this built-in redundancy for handling X chromosomes that it just doesn't have for autosomes.
Okay, so non-disjunction covers errors in chromosome number. But what about errors in chromosome structure? You mentioned earlier that faulty crossing over or repair in prophase could cause those.
Yes, absolutely. The mechanics of breaking and rejoining DNA during recombination are complex, and mistakes can happen. We see errors like deletions, where a whole segment of a chromosome just gets lost.
Like a piece just snaps off and disappears.
Essentially, a classic example from the sources is the 5-toler deletion, a missing piece in the short arm, the P arm, of chromosome 5. This causes Cri-du-Chat syndrome.
Cri-du-Chat, cat cry.
Exactly. The most distinctive feature is this high-pitched, cat-like cry in affected infants, along with microcephaly and developmental delays. Losing those specific genes on that missing piece disrupts normal development.
And the opposite can happen too, duplications.
Yes, duplications, where a segment gets accidentally copied and inserted so you have extra copies of some genes. These often lead to developmental delay as well, again due to gene dosage issues. But perhaps the most disruptive structural errors in terms of fertility are things like inversions and translocations.
How do those work?
An inversion is when a segment breaks off, flips 180 degrees, and reinserts itself. A translocation is when a piece breaks off one chromosome and attaches to a completely different, non-homologous chromosome.
Do these always cause problems for the person carrying them?
Not necessarily for the carrier themselves. If no genetic material was actually lost or gained, they might be perfectly healthy. The problem arises when they try to have children, when their cells enter meiosis the first.
Because the chromosomes won't match up properly?
Exactly.
Yeah.
Imagine trying to pair up a normal chromosome with one that has a flipped segment or a piece stuck on from somewhere else. Forming that synaptonomal complex becomes incredibly difficult, if not impossible.
The zipper gets stuck.
The zipper gets stuck or can't form correctly. This difficulty in pairing during prophase eyes leads directly to a very high chance of producing gametes that are aneuploid missing or having extra pieces of chromosomes. That's why balanced translocations and inversions are a major cause of recurrent miscarriage and infertility.
Makes sense. And finally, we have those really small errors, the point mutations, single letter changes in the DNA code.
Yes, these often arise from mistakes during DNA replication just before meiosis, or potentially from errors in the repair process during recombination. And they lead to many of the classic single-gene inherited disorders.
Like sickle cell.
Sickle cell anemia is a perfect example. It comes down to a single point mutation in the HBB gene, which codes for part of hemoglobin. Just one DNA-based change swaps one amino acid, glutamate, for another.
Such a tiny change.
Tiny change, massive consequence. This alters the hemoglobin protein, causing red blood cells to distort into that characteristic sickle shape, especially under low oxygen conditions. This leads to chronic anemia, pain crises from blocked blood vessels, vaso-occlusion, and organ damage.
What about cystic fibrosis?
Cystic fibrosis is another key example. The most common mutation, called delta 508Rs, is actually a deletion of just 3 DNA base pairs in the CFTR gene. This tiny deletion, often thought to arise from errors when replicating repetitive DNA sequences, causes the resulting CFTR protein to misfold and get degraded.
And that protein is important for?
It's a chloride channel, crucial for maintaining the right balance of salt and water across membranes, especially in the lungs and digestive system. Without it, you get that thick, sticky mucus buildup, leading to chronic lung infections and digestive problems.
And the thalassemias.
Thalassemias are a group of disorders affecting hemoglobin production, similar to sickle cell, but often caused by larger deletions or faulty gene repair affecting the globin genes during meiosis. This leads to reduced production of hemoglobin chains, resulting in microcytic anemia, small, pale red blood cells.
Okay, so we've covered number errors, structural errors, point mutations, but the sources also mention things beyond the sequence itself, right? Epigenetic errors and links to more complex conditions.
Yes, it's not just about the DNA sequence. Imprinting errors are a fascinating example. Imprinting is a process where certain genes are chemically tagged, usually by methylation, in the sperm or egg cells during meiosis. These tags essentially dictate whether the gene inherited from the father or the gene inherited from the mother will be switched on in the offspring.
So it's like an instruction tag applied during gamete formation.
Exactly. And if the tagging process goes wrong during meiosis for genes on, say, chromosome 15, you can get dramatically different diseases depending on which parent's gene had the faulty imprint. Errors can lead to Prader-Willi syndrome, often associated with weak muscle tone in infancy and later uncontrollable appetite leading to obesity. Or, if the error affects the other parental copy, it can cause Angelman syndrome, characterized by severe developmental delay, seizures, and often a happy demeanor with frequent laughter. Same region of DNA, different parental origin of the error, different outcome.
Wow, that's complex.
And furthermore, there's growing evidence linking subtle structural changes that can arise during faulty recombination things called copy number variations, or CNVs, to susceptibility for highly complex polygenic conditions.
CNVs. Like small deletions or duplications.
Yes, often too small to be detected by older methods. These CNVs, essentially born from minor errors in meiosis, don't cause conditions like autism spectrum disorder, ADHD, schizophrenia, or bipolar disorder on their own, but they seem to act as risk factors, potentially predisposing individuals by slightly altering the dosage of multiple genes involved in brain development and function.
So the fidelity of meiosis has ripples that affect not just obvious physical syndromes, but potentially the very wiring of our brains.
It really underscores how fundamental this process is to almost every aspect of health and development. Hashtag take hats, broader relevance in the case of keratoconus.
This profound influence of meiosis clearly extends way beyond just fertilization and early development. Let's broaden the view a bit now, thinking about clinical relevance later in life and also the bigger evolutionary picture.
Okay. A really crucial and heavily studied clinical aspect, which we touched on briefly, is the maternal age effect.
Right, the increased risk of certain chromosome problems with older mothers.
Exactly. We know definitively that the risk of non-disjunction, especially leading to trisomy 21 Down syndrome, rises quite dramatically as maternal age increases, particularly after age 35. And the sources clearly link this to the biology of the female gametes, the oocytes.
Why is that? What's happening in those older oocytes?
Well, female oocytes start meiosis before birth, but then they arrest. They pause right in the middle of prophase, that most complex stage with the paired chromosomes and synaptonomal complex.
And they just stay there. For years, decades.
For decades, potentially. They stay arrested in prophase from fetal development until ovulation, which could be 15, 25, 35, even 45 years later. It's an incredible feat for a cell to maintain that state for so long.
That sounds like a huge demand on the cell's machinery.
It is, and the cellular machinery, specifically the protein structures holding things together, appear to degrade over time. The maternal age effect is thought to be directly linked to progressive cohesin decay in those arrested oocytes.
Ah, cohesin again. The molecular handcuffs holding sister chromatids together.
Precisely. Those cohesin rings, especially the ones at the centromere being protected by Shugoshin, seem to slowly break down or become less stable over those decades of arrest.
So they get weaker.
They get weaker. Then, when the oocyte finally gets the signal to resume meiosis decades later, right before ovulation, those weakened connections make the chromosomes more susceptible to being pulled apart incorrectly. The sister chromatids might separate prematurely, or the homologs might not disjoin properly. This increases the chance of non-disjunction, leading to an aneuploid egg.
So time itself degrades the machinery needed for accurate separation.
That seems to be the prevailing model, yes. And interestingly, the relevance of meiotic processes extends even into areas like cancer research.
How so?
Well, our sources note that many genes that are normally only active during meiosis are found to be aberrantly switched on in various types of tumors.
Really? Meiosis genes in cancer cells?
Yeah. And their inappropriate activity seems to contribute to the widespread genomic instability, the chaotic chromosome numbers and structures. That's a classic hallmark of cancer. So understanding the normal checks and balances that control meiosis might give us insights into what goes wrong when cell division runs amok in oncology.
That's a fascinating connection. Thankfully, our ability to actually see these meiotic errors has improved dramatically, hasn't it? We're not just relying on looking at chromosomes under a microscope anymore.
Oh, absolutely. While the basic karyotype is still fundamental, modern genomic medicine uses a whole toolkit. Fluorescence in situ hybridization, FIH, uses fluorescent probes to light up specific DNA sequences, allowing us to count chromosomes or spot translocations. Array comparative genomic hybridization, array CGH, is brilliant for detecting tiny deletions or duplications, those CNVs we mentioned, across the entire genome by comparing patient DNA to a reference sample. And increasingly, we're using high-resolution whole genome sequencing, WGS, to map out not just number and structure, but the exact sequence and even epigenetic modifications, giving us an incredibly detailed picture of meiotic outcomes.
The technology has really caught up to the complexity. Now, if we pull back even further to the evolutionary scale, why does life even bother with something as complex and error-prone as meiosis and sexual reproduction? Asexual reproduction seems so much simpler and less costly.
That's the multi-billion-dollar evolutionary question, isn't it? And you're right, meiosis is energetically expensive. The sources mentioned the ATP needed to power the spindle movements, the enzymes for recombination, the motor proteins. It takes a lot of cellular energy.
So why pay the price?
The overwhelming consensus is that the benefit of the genetic variation generated is just too valuable to pass up. That novelty created by crossing over an independent assortment allows populations to adapt much more quickly to changing environments, parasites, diseases, you name it.
So the variation is worth the cost and the risk of errors.
It seems so.
Yeah.
There's this concept mentioned in the notes, emergent order. It's the idea that out of the seeming chaos of random assortment and recombination, a beautiful functional harmony emerges, this balanced diversity that fuels evolution. Life uses controlled error, in a way, as a source of innovation.
Controlled chaos yielding harmony. I like that. Okay, to bring this huge topic back down to a specific clinical example that illustrates how meiotic fidelity can impact health much later in life, let's talk about the case study in our sources.
Keratoconus.
Right, keratoconus is a really good example of complex inheritance potentially linked back to meiosis. It's an eye disorder where the cornea, the clear front window of the eye, progressively thins and starts to bulge outwards into a cone shape.
Instead of being a nice smooth dome. Exactly. And this cone shape distorts vision, causing significant nearsightedness, myopia, and irregular astigmatism that often can't be fully corrected with regular glasses or contacts.
Is it common?
It's relatively common for a genetic eye disorder. The prevalence data in the sources varies by population, but suggests it affects somewhere between, say, 55 per 100,000 people of white European descent, up to maybe 229 per 100,000 in some Asian populations. So not incredibly rare.
And the sources emphasize a strong genetic component, even though environmental factors like eye rubbing are involved.
Absolutely. While things like chronic eye rubbing or oxidative stress might trigger or worsen it, the genetic link is undeniable. Your risk of developing keratoconus is estimated to be 15 to 67 times higher if you have a first-degree relative with the condition.
That's a huge increase in risk. So how does meiosis tie into this? It's not a simple single gene thing, is it?
No, it's definitely not. It's considered a complex, multifactorial disease, likely polygenic, meaning multiple genes contribute small amounts to the overall risk. The specific meiotic link is thought to be related to the inheritance of combinations of these susceptibility alleles.
So getting an unlucky combination through independent assortment.
That's one strong possibility. Or, potentially, subtle copy number variations, CNVs, affecting relevant genes could arise from minor meiotic missegregation events, contributing to risk.
Are specific genes known?
Several candidate genes have been implicated, many involved in collagen structure and corneal biomechanics. The sources mention genes like DOCK9, LOX, which is involved in collagen cross-linking crucial for corneal strength, a microRNA called MIR184, and FNDC3B, among others.
So these genes related to corneal structure get transmitted through the germline via meiosis.
Right, and either a faulty recombination event involving one of these genes or simply inheriting an unfortunate combination of several risk versions of these genes due to the random shuffling of meiosis could set the stage for the cornea to weaken later in life.
So it's a condition that might not show up until adolescence or adulthood, but its origins could trace all the way back to the meiotic events that created the sperm or egg cell decades earlier.
Exactly. It perfectly illustrates that long ripple effect, how the fidelity of those molecular events during gamete formation can influence susceptibility to a very specific tissue failure, in this case the corneal matrix, much, much later. Hashtag tag tag outro.
Well, that brings us to the end of this particular deep dive into meiosis, the incredible engine of genetic diversity. We've really unpacked how this remarkable process uses two intricate divisions to achieve both massive complexity and that essential reduction in chromosome number. Leveraging things like crossing over and independent assortment for variation, all while relying on those master molecular clocks like the cyclin CDK complexes and those absolutely critical integrity checkpoints involving proteins like P53.
Yeah, and what really remained striking, I think, after reviewing all this, is the inherent fragility of the system we've just described. We spent a lot of time on the elegance of the molecular dance, but also its vulnerability, especially, as we discussed, that strong link between maternal age and the decay of cohesin in those long-arrested oocytes. It's such a clear pathway to non-disjunction.
Right. The very system that creates such amazing diversity is constantly operating right on the edge of potential failure.
It truly is.
Which leaves us with this final provocative thought for you, our listener, to perhaps chew on. We established pretty clearly that cohesin decay over time seems linked to that increased risk of non-disjunction as female gametes age. Now, considering the source notes highlight that the protein shigoshin, the guardian angel, is absolutely required to preserve that critical centromeric cohesin during meiosis the first, how critical, then, is the precise timing and functional efficiency of shigoshin itself, perhaps very early in development when oocytes are first forming? And maybe more intriguingly, what unexplored environmental factors, or perhaps internal cellular factors, might impact shigoshin's effectiveness over those long decades of arrest, potentially altering the genetic landscape of that egg cell before life even officially begins?